that Wednesday is your exam. Okay? Is that Pepto or is that strawberry milk?
It's just strawberry.
Okay. So, oh gosh, she's telling me, really, Olivia? You're that sick? You're gonna like shake it up, Pepto, and no. All right. So remind your exams on Wednesday on adaptive immunity and the respiratory system. If you have any questions, let me know by email. I will be in the office from about 9:30 to 11 tomorrow as well. If you want to stop by before lab courses for the afternoon. All right, so today we're going to finish up digestion. Okay, we're going to finish up digestion today. So we went through mechanical digestion in the mouth, the oral cavity, esophagus, what happens in the stomach, special anatomy of the stomach, what in terms of chemical digestion happens in the mouth and in the stomach, mechanical digestion in the stomach, and we left off talking about the liver and the gallbladder. All right, I want to go over the gallbladder today. Gallbladder sits in this little indentation on the inferior surface of the liver, and the purpose of the gallbladder is to store what? Store and release bile. Right? It releases bile through the cystic duct, eventually into the common bile duct, and that then proceeds down to the to fuse with the pancreatic duct and empties into the duodenum of the small intestine. Produces and stores bile. What's the major problem that happens with the gallbladder? Gallstones. Okay. Gallstones. And so when you get a gallstone, right, the bile, which is a salty, like salt-filled solution, it calcifies, it hardens, and it blocks this, right? Either blocks the release of bile into the common bile duct, or it actually can block the bile duct itself. Okay? And so what ends up happening is they have to have a surgery to remove the gallbladder. Okay? You still produce bile from the liver, and it just empties into the small intestine without storing it. Okay? So gallstones. And it could be a pretty debilitating, painful condition. It could also causes problems with digestion and problems with diarrhea and stomach discomfort in general. Okay? The other accessory structure we need to talk about is the pancreas. Okay? The pancreas is retroperitoneal. It sits behind the stomach. Okay? And it has a certain structure, right? So we have the head is this area closest to the duodenum, the body, right, as it gets smaller, and then the tail, this smaller piece that's further away from the small intestine, closer to the spleen. Okay? The connective tissue of the pancreas actually divides this into what we call lobules. All right, so lobules. So you see this, it looks like, when you look at a pancreas, if you've ever seen one, you can't really see it well, you can see it a little bit in this cadaver picture. It's actually kind of, it looks like it's, how do I want to say it? It looks like it's got this like furry type of texture to it, right? In a live specimen, it kind of looks like it's kind of like bumpy, like it's kind of like shag carpeting, okay? And because the way that the connective tissue separates it into different lobules, right? And then it kind of moves around. It's what we call this amorphous structure, right? I was doing some post-op work on pancreatic cancer cells, and I'd have to inject them into the pancreatic cancer cells into the pancreas of a mouse. And you try to like stab this thing, and the pancreas like moves. It's really hard to do, right? It's kind of like, anyways. And the pancreas of a mouse is like that big. So you have the pancreas. Pancreas is made up of a multitude of cells, right? And each of these lobules actually is composed of what we call acini, groups of cells, right, that are lobbed together. And the pancreas has two distinct functions, right? Most of the acinar cells, right, the acinus is really this gland that looks something like this. okay? And there's cells that are around it that form this gland, okay? Each of these cells, and they have nuclei, and there's different types of cells that form this acinus, and they produce proteins that are released into the lumen. All right, so they're released into this area right here, and then it exits out, eventually getting together into the pancreatic duct. Okay, so we have acinus, and then everything. You can see the duct. So you say whatever's produced actually gets down here, then collected through this way, and then out into the duodenum. All right. So these acini, groups of cells, and there's a multitude of different types of cells that are present in the acini. They produce the stuff that makes up the exocrine function of the pancreas. Exocrine. Okay? Meaning that the secretion, what they're going to produce, is actually going to end up out of the body. What are most of the acinar cells, what are they producing? They're going to produce enzymes, right? Enzymes and pancreatic secretions, collectively called pancreatic juice. Yeah, that's good. That's great right after lunch, right? Pancreatic juice. And that's a gem. Okay, you can see here all of the secretions are collected into this pancreatic duct, and there's the main pancreatic duct, and then there's an accessory one, but both of them terminate into the duodenum where the pancreatic enzymes and pancreatic juice will be released. Okay? That is the exocrine function. What do we know about the other function of the pancreas? Pancreas also has an endocrine function. What are those? What's the endocrine function of the pancreas? To do what? Insulin. And what does insulin do? Regulate blood sugar. So really we're going to regulate blood sugar. Okay? And this is a whole different part of the pancreas. This is where we have the islets. Right? You saw those in lab. Islets of Langerhans, or what we call pancreatic islets. Okay, so if you had tissue of the pancreas that kind of looks like this, actually makes that a little bit bigger. So, all right, so if all of these were the acinar cells, inside the acinar, like separated, are these areas. They kind of look like little circly, squiggly things when you're looking at them under the microscope. Okay, those are your pancreatic islets, and they contain beta cells and alpha cells. What do the beta cells produce? I forget how the Savior's whistle goes in Walking Dead. Beta cells produce what? Insulin. Okay? Insulin. And alpha cells produce its opposite hormone, glucagon. Okay? Glucagon. So both of these cells are present in the islets. They produce hormones that are released in the blood which help regulate blood sugar. Okay? Insulin will be released in response to high blood sugar to help lower it, and glucagon will be released in cases of low blood sugar to help raise it. Right? And that's the endocrine function of the pancreas. Okay? Questions on that? And we cover out the endocrine function. All right, so pancreatic juice is that secretion. It's formed from the acinar cells, right? And some pancreatic duct cells that are within there, okay? And it is made up of an alkaline fluid. What does alkaline mean? pH right, less than, or let's actually call it greater than 7, right? It's basic. Why is it basic? It's mostly water. It has some of this bicarbonate ion and some digestive enzymes. That's what makes up this pancreatic juice. But why is it alkaline? Is it important that it's alkaline? If it wasn't, I wouldn't be mentioning it, right? Why is it alkaline? Why is it basic? Olivia?
Because everything in the stomach, coming out of the stomach, is really acidic.
Exactly. Okay, so the kind that's leaving the stomach, right? Okay, we have the stomach here, right? And then we get to the pylorus. This is the stomach. Okay, and then we come to the duodenum right here. Okay, so kind that's leaving the stomach this is very low pH. So the food that's leaving the stomach is very low pH. And now the pancreas is releasing all of these enzymes, amylase, lipase, proteases, nucleases. They're going to break down molecules. Okay? They're optimized to work at what pH? You think they work on pH 1? What are these enzymes? What are they? What type of molecule are they? Proteins. Do proteins have an optimal pH that they work at? Yes. Okay? So this helps to regulate, right, and neutralize the pH of the chyme as it's coming out of the stomach so that these enzymes can work. Okay? And it's up to right now, where have the enzymes, what enzymes have we covered? And where do they come from? We covered what? Amylase. And where was that found?
Salivary gland.
All right, salivary gland. What else? Lipase. We had a lipase that came from where? Also from the saliva. Okay, where else did chemical digestion occur so far? Stomach. What happened in the stomach? Protein broke down through the action of what enzyme? Pepsin. Okay? So pepsin. But so far we haven't, have we done much digestion? Chemical digestion? No. Okay? So the bulk of the chemical digestion that occurs and all of the absorption actually occurs in the small intestine. Okay? So, and it's aided primarily from the pancreas. Release of amylase, lipases, proteases, nucleases, all these enzymes are going to start to work on mix with the chyme that's released from the stomach as it gets into the duodenum, or duodenum, however you want to call it. Okay? And they're mixed, and then now we have digestion that's occurring. Remember that, okay? Here's an example or a picture of the pancreatic islets versus the acinar cells. All right, here's a acinus right here, this whole thing, and it's attached to a duct. Okay, you actually see the duct leave here. Okay, and the acinar cells are there. Different than the structure of the pancreatic islet. Okay, the pancreatic islet has alpha and beta cells and blood vessels. Okay, out here there's less blood vessels. Right, you need blood vessels in there because it's going to produce hormone that are going to be released into the blood. Okay, and you have acinar cells. That acinar cells produce the enzymes. The duct cells help produce the bicarbonate, which regulates pH. Right, together they produce this pancreatic juice that's going to be released into the duodenum, stimulated by the presence of chyme leaving the stomach and entering the small intestine. Questions? So how is digestion regulated? All right, so we went through, I want to go through this a little bit so that you understand there's a relationship about how and when, as food passes from stomach to different parts of the canal, the gastrointestinal tract, that there's a stimulation that occurs to prepare the next step, all right, and inhibit the step previous, okay? And so there's some hormones that are involved. We already talked about one. We mentioned one with the stomach. What was it? What's the stomach hormone we talked about? Gastrin, right? Gastrin. It helps regulate the motility of the stomach, okay? When it's released, it's secreted in response to that, the filling of the stomach. It increases motility, okay? And there's other ones, two others. Cholecystokinin, or CCK, okay? And secretin. They're both released in response to chyme in the food, presence of chyme in the small intestine, okay? Cholecystokinin is released from the small intestine in response to fatty chyme. So you go out and you have a nice plate of buffalo wings, greasy buffalo wings, right? And that chyme then sits and is digested, and there's a lot of like fat content. That then goes to the small intestine, and it regulates, there's receptors in there that stimulate the release of cholecystokinin. CCK stimulates the gallbladder to release bile, and then stimulates the pancreas to release pancreatic juice. Okay? So release bile, why is bile important? What is this in response to? What type of chyme? Fatty acids. Right? So if fats are present, we need bile to help digest those fats. Okay, so it stimulates the release of bile. Okay, and then it inhibits stomach motility and release of gastric secretions. Food is moving into the small intestine. The chyme is moving into the small intestine. We don't want to push more. Let us digest this first. Slow down the motility of the stomach, which would produce or push more chyme into the small intestine. So it's a stepwise process. A little bit, hold up, we're gonna send the signal back, slow down, we're gonna digest this, and we're gonna move on. All right, secretin, another hormone is released in response to chyme acidity. Chyme acidity. What is chyme coming from the stomach high in pH or low in pH? Colin. Low, right? So it's gonna be acidic. So as soon as it gets there, the secretin is going to be released, and that's going to stimulate the release of the pancreatic juice, that is the alkaline fluid that was going to help neutralize that acidic chyme. And then both of these feedback on the stomach to slow things down, to allow the small intestine time to help digest the material that's coming from the stomach. All right? So here's a little table, talk about the hormones and what it does. Gastrin, cholecystokinin, and secretin, and what they do. Okay? And where they come from. We're talking about G cells in the stomach, enteroendocrine cells in the small intestine, and where things are. So the food gets into the stomach, that's going to stimulate motility. Gas is going to be released, stimulate motility, mechanical digestion, mixing, release of pepsinogen, etc. Right? So we want the enzymes there. Now food moves to the small intestine, cholecystokinin and secretin are going to be released. One, to neutralize the pH. One, to release enzymes, right? And also to release bile from the gallbladder. Right? Presence of the material that we're going to digest, we need the enzymes and the material that's going to do that digestion there at the same time. Okay? Intuitively based approach. Okay? And what do those enzymes do? Now that they're present and mixed with the chyme, what are they going to do? I'll wait. What are they going to do? What are the enzymes going to do? They're going to sit there, like you guys, bumps on logs. Emma! They're going to break down the food you consumed, okay? We have to break them down. We have to break food down into absorbable nutrients. We consume food in the form of macromolecules. Last semester we went over the structure of macromolecules and what they are. Carbohydrates, proteins, lipids, nucleic acids. We talked about how they are built up from smaller subunits. Now we're breaking them down into smaller subunits in order for them to be absorbed into the bloodstream. Okay? So here's a breakdown of the digestion of molecules and where it occurs. Carbohydrates, proteins, lipids, and nucleic acids. Okay? Oral cavity, saliva. All right? Starch and some lipids. In the stomach, proteins are broken down. Okay? There's some lipid digestion that occurs. But then in the small intestine, look at this. Here we have the enzymes of the pancreas, break down starch and other carbohydrates, proteins, lipids, and nucleic acids. And then finally the rest of stuff, the brush border enzymes. What's the brush border of the small intestine? Villi and the microvilli, okay, that sit on top of those circular folds, right? The microvilli, increased surface area, that's the brush border. And those cells at the brush border produce enzymes, right? Maltase, lactase, sucrase, dipeptidase, aminopeptidase. They're going to break down carbohydrates, proteins, nucleic acids, right? And there's no breakdown of lipids in this case. Okay? So that now the digestion is completed, and now what happens? Now we have these small subunits, amino acids, monosaccharides, we have fatty acids and glycerides that are present, nucleotides. They get absorbed into the bloodstream. Okay? So the digestion of macromolecules. All right, so all of this happens in the small intestine. So we talk about pancreatic amylase, comes from where? Abby.
Where is it produced?
Yes.
Pancreas.
Okay. And what does it break down? Amylase breaks down?
Starch.
Okay. So this is the breakdown of starch. Starch is a polysaccharide made up of a line or a complex of glucose molecules. Pancreatic amylase is going to enter at the duodenum. The starch is already partially digested from the saliva. Okay? It's going to be broken down and go from there. We have others, so pancreatic enzymes, continuous digestion of starch, and the brush border enzymes will break down the rest of the other polysaccharides and the disaccharides that are present. Lactose, maltose, etc. Then those carbohydrates are absorbed into the at the small intestine into the bloodstream. And then that blood goes where? Where does the blood from the small intestine go after absorption of molecules? Goes to the tissue. What tissue? No. Of carbohydrates, when carbohydrates gets absorbed to the blood, where does that blood go? From the small intestine through the inferior and superior mesenteric artery vein to where? Think again. Doesn't go to the kidneys. Look it up. Go, look it up. I'll give you 30 seconds. If you go back, it's Joe. Wait. Joe has the answer. The hepatic portal vein. Danny didn't say it. Okay? If you're like, oh, I got it, I knew it. Well, what? Everyone's quiet. I'm assuming you don't know if everyone's quiet. Okay? Hepatic portal vein, which then goes to the liver. Awesome. Then the liver, we go to the sinusoids,
So why
right?
is the liver wrong? Doesn't that go, so it goes to the liver?
Yes. Who said it was wrong?
You did.
When?
We just said liver, kidney, and...
No, I did not hear liver. I heard kidney.
Oh, okay.
I did not hear liver.
You said wrong. Think again.
No, I heard Lily say kidney.
No, you said liver. You were like,
No, I definitely did not say that. So, all right, so breakdown of this occurs.
Right answer.
Digestion of starch. Right, so starch is this long chain of glucose molecules, and it gets broken down partially by salivary amylase, okay, partially by pancreatic amylase, and then the rest of the stuff, dextrinase, which dextrin is a long chain carbohydrate as well, and glucoamylase. They break the bond between glucose subunits of oligosaccharides. Right, so starch broken down into these kind of chunks and some disaccharides, and then the rest of it break it down. What do you end up with? What's the end point of starch digestion? What's the end?
Glucose.
Glucose molecules. That is what is absorbable into the blood. Blood that goes to the liver from the hepatic portal vein. Okay, okay. Digestion of other disaccharides, right? So amylase breaks things down, these disaccharides, lactose, you need this enzyme lactase, sucrose, sucrase, and you go on, right? And eventually you end up with monosaccharides. The end point of carbohydrate digestion are monosaccharides. Okay? Galactose, glucose, fructose, those are all monosaccharides, including pentose, ribose, deoxyribose are also, that's the endpoint, partial endpoint of nucleic acid digestion. Okay? Proteins are digested also in the small intestine. So we have partially digested proteins that enter the duodenum from the stomach, okay, and then they're going to be proteolytically digested by pancreatic enzymes and brush border enzymes. And what they do, okay, there's a couple important ones, okay, trypsinogen and chymotrypsinogen. What do they sound? What is nogen? What does that sound like? Synogen. What does it sound like? Pepsinogen. What was pepsinogen? Emma, was it an inactive form of pepsin? So what do you think these are? Inactive forms. Okay? They need to be activated. They're activated based upon cleaving pieces of a part. They're activated to trypsin and chymotrypsin. Okay? And trypsin breaks down a bunch of different proteins. Okay, we're going to go over those in a second. Okay? And so you see here, trypsin and chymotrypsin recognize certain bonds, certain peptide bonds within proteins. All right, unique ones between different amino acids. All right, and aminopeptidase also targets those peptide bonds. Okay, so protein. What's the end point of protein digestion? The end point of carbohydrate digestion was what? Specifically monosaccharides, right? Because we also have galactose and fructose. And the end point of protein digestion is going to be what? Amino acids. Okay? And so you break these down. As it leaves the stomach, it's broken into from complex macromolecules to less and less complex, eventually getting to the brush border enzymes that break them into final amino acids, which then can be absorbed into the bloodstream and brought to the, all together now, liver, all right, for lipid digestion. Okay? Now lipids are a little bit different. Okay? Because lipids are what? What is one of the hallmark characteristics of a lipid? That's what it is, not what it.
Hydro.
They're hydrophobic, right? Hydrophobic. And this, the chyme at this point is a watery substance, has lots of water in it, okay? So when you get fat that's in there, it's clumped together. It's formed a mycel with something that looks like this, all right? Where the fat droplet is surrounded by water molecules, okay? And what we want to do is these bile salts, bile itself helps emulsify that and break this up, okay? It interferes and surrounds the lipid molecule so it can't clump together with other lipid molecules, okay? And allows the enzymes, pancreatic lipase and other enzymes of the brush border to break things down. So pancreatic lipase will take a triglyceride, which is a fat, a glycerol molecule with 3 fatty acids, and break it into a monoglyceride. One glycerol molecule with 1 fatty acid and 3 fatty acids that will be absorbed. Okay? Now the absorption of these fat molecules and these broken down fatty acids is different. It does not go into the blood. Okay? That causes problems. When you have a high fat diet, you would look, you go to someone has a high fat diet, might end up with cardiovascular disease, arteriosclerosis, because they have triglycerides in the blood, high triglycerides, which cause problems in fatty blood vessels. Right? So if there's too much here, you overcompensate for this. So you end up with the absorption of the fat molecules across the cell. You get these chylomicrons, right, that are produced, right, which is a protein bound to a lipid carrying molecule across the cell, and then the lipids are absorbed into the lacteals of the villi. Okay, that is the part of the lymphatic system. Okay, and they help package and transport the fats into the lacteals so that they can be absorbed. So the bulk of digestion, whether it be carbohydrates, proteins, or lipids, occurs in the small intestine, okay, with help from the pancreas and the liver and the gallbladder. Questions on that? We're not really going to cover the breakdown of nucleic acids, okay, that basically occurs in the small intestine the similar way that we have here. Okay? So now we've absorbed most of our nutrients. We've produced and digested most of the material. Most of the material that's left is undigestible. Okay, most of it. And then it moves from the small intestine, from the ileocecal valve, into the large intestine, otherwise known as the colon. Okay, the first part of the large intestine is the cecum, ileocecal valve. So chyme moves through here, empties into the cecum, kind of a holding area. All right, and we will go through the different parts of the large intestine. You have the ascending colon, the transverse colon, the descending colon, the sigmoid colon, the rectum, and the anal canal. That is the end of digestion. Okay? So at this point, up to this point, 95% of digestion has occurred. Okay? Within the large intestine, now, this is like the water reclamation facility. Okay? We need to take back all the stuff, some of the stuff we put in. We put a lot of water into an effort into digesting this food. Okay? And so now we're going to take some things back and get back as much water out of the food as possible to help retain it. Okay? So food comes into the cecum, and then it is moved through the large intestine via peristalsis. Okay? Peristalsis in the large intestine is slow. Right? It slows down. Allows time for the cells to reclaim the water that could be lost. All right, so some other anatomical parts. All right, so there's a turn. Each of the turns are called flexures. So ascending colon, there's a right turn, the right colon flexure, okay, on the right side of the body. On the left side of the body, there's a left colon flexure that it turns down to the descending colon, and then a sigmoid flexure where the sigmoid colon is. Okay? You'll note that there's this kind of bumpy appearance to the large intestine. Each of those bumps or bulges is called a haustra. Haustra, plural. Okay? The way this is, the way that the intestine is and how it's structured, the muscle bulges a little bit, and that's what gives it this kind of rounded appearance. There's also a ligament that holds all this together called a tenei coli. Okay, questions on that? At the end, you have the rectum and the anal canal. Right? The point of this, all of this colon is to help hold fiber or hold waste so that we can reclaim as much stuff out of it as possible. Okay? There's also a lot of bacteria that live in the colon. The bacteria, what we call, they are essential microbiota that are present. They will help further break down some of this food. Okay? They'll produce B complex vitamins for you, which will be absorbed. And then they'll also produce vitamin K, which will also be absorbed. All right, but we need those bacteria. We don't do that. That's part of them, and they provide that for us. We provide them a food source. They provide those materials for us. Okay? So it absorbs water and electrolytes. That chyme is compacted into feces. All right? It stores feces until eliminated through defecation. Okay? Help reclaim as much water as possible. Then we have the indigenous microbiota. All right, those break down carbohydrates, proteins, and lipids that are remaining there. All right, and produce carbon dioxide, gas. All right, hydrogen ions, some other substances, including methane. All right, B vitamins, vitamin K. The final product is waste, fecal material, including water salts, some epithelial cells, bacteria, undigested material, fiber. Along the walls of the large intestine are these what we call intestinal glands. All right, they're covered with simple columnar epithelial tissue, similar to what you see in the small intestine, but there's a lot more goblet cells that are present. So we still have mucosa, submucosa, muscularis, and the serosa layers. Right, there's some lymph nodules. Right, and then you have this intestinal gland that's present. Okay, and you'll notice the large amount of goblet cells that are present. They produce mucus. Mucus helps line the large intestine, right, so that the waste can easily pass through, so it doesn't get stuck. Okay, so we said that peristalsis was weak, sluggish, it's slow for a reason, so that we can reclaim the water and allow the bacteria to produce those B complex vitamins, etc. And each haustra churns. Okay, so there's a movement, there's a contraction in one area, relaxation in the other, that helps move the waste material from one area to the next. A relaxed haustrum will be filled with material, right, from the previous haustrum that's churning through. Right, so it helps move material one area to another. Okay, and the tenei coli help with some powerful contractions, which help propel the fecal material towards the rectum. Okay, and eventually this happens 2 to 3 times a day, often after a meal. Okay, this responds to the filling of the stomach. right, to keep moving material through the body. Okay, that's what's called the gastrocolic reflex. Okay, the stomach distends after a meal. Right, you have a meal, stretch of the stomach, that causes a mass movement, initiates a mass movement of the colon as well. Okay, there's a reason why my dog needs to go outside after he eats. Just the way it is. Okay? And then the other part of elimination of waste is the defecation reflex. Okay? As the rectum fills with waste material, there's an urge to eliminate that waste. Okay? The stretch of the rectum pushes pressure on nerves of the sacrum, and there's a response to the spinal cord. Right, and then there's an increased parasympathetic output to the colon, right, and decreases output to internal. Eventually, defecation is not only a reflex, but also a voluntary activity. Okay, so part of it is a reflex action, right? Digestion, elimination, parasympathetic input causes the sigmoid colon and the rectum to push material into the anal canal, and then we have to relax the voluntary muscles that will allow defecation to occur. Okay, so it decreases the output to the involuntary anal sphincter, a muscle that holds it in. Okay, and then you have to relax the external anal sphincter in order to allow material to be eliminated from the body. So the voluntary defecations learned about age 3, right? They understand what that feeling is, that spinal cord reflex, and what that means to do. Okay. Questions on the large intestine. Okay, so this is the defecation reflex. Right, rectum content stimulate, stretch in the rectal wall. That sensory input goes to the spinal cord. Then that's delayed. Those parasympathetic motor neurons go to more increase here, relax the internal anal sphincter, and then you have to go somatic neurons to relax the external anal sphincter, which allows passage of material out of the body. Questions on the large intestine or defecation. Sometimes we end up with problems with the large intestine. One of the biggest problems is colon cancer. Okay, causes polyps that arise within the colon. All right, and that happens over years and years of mutations that occur. All right, some other problems include problems with the bulges of the small intestine. Okay. Diverticulosis and diverticulitis, okay, are 2 examples. This is the large intestine, and some places it bulges out. The haustra bulge out because there's a narrowing of the colon and there's low fiber. The bulge occurs, a loss of the muscle becomes slow or weak, and you end up with a bulge in the large intestinal lining. What happens there? Material gets stuck in there in some places. And sometimes, so diverticulosis is a pretty benign type of condition. It's present, okay? But when you end up with a inflammation of the diverticulosis or diverticula, the bulges, that can cause big problems, okay? So inflammation occurs, causes pain, stomach discomfort, causes colonic spasms, it causes diarrhea, all right, and then you leak if a rupture of this occurs, right, the bacteria that are present within the colon are now exposed to the abdominal cavity. And that can cause sepsis, right, and you'd be a life-threatening situation. Okay, so then other important parts of this, well, important conditions of the large intestine: constipation, diarrhea. Constipation, impaired ability to defecate. The compacted feces become less and less water, right? Which that of water content make them more and more difficult to eliminate, right? So this could be dehydration, lack of exercise, poor bowel movements, anesthesia, which slows down bowel movements, a couple other things, medications. And then diarrhea is quite the opposite: disruption in the normal mechanism, right, to absorb intestinal water for whatever reason. Amoeba, colonic dysentery that occurs. The stool, the feces has a high water percentage, water content, and it's difficult to deal with. All right. So, question: before you leave, you're gonna have this exit ticket for Socrative, and you're gonna answer that question. That covers and completes, digestion, right, of the digestive system. Yeah, that's the third one. This is just number 3. What, Alexa? That's what I thought. That's not the answer for number 2, is it? Nothing? What you learned in class today? Nothing? Remember, on Wednesday, we'll have our exam.